So for those of you who do not know, I am Charmaine Dean. I am the Vice President Research at the University of Waterloo. It's such a privilege to serve here in that capacity. I'll also take the opportunity right away to introduce Bernie Dunker, who is an Associate Vice President, and his portfolio is interdisciplinary research, and he'll be taking the question and answer period at the end. Research Talks is a regular series that we offer, and we have been moving it into the realm of cross-disciplinary, interdisciplinary research discussions. And we are very fortunate to have a wonderful panel here today to talk about innovative approaches to revolutionizing healthcare. The session was organized by Associate Vice President John Thompson, who's been doing superb work on making sure that we have an interdisciplinary flavor to the research talks since I arrived. So I'd like to tell you a little bit about the researchers who form our panel, and then they'll have a few minutes to give us an overview of their presentation, of their work, and then subsequent to that, we'll bring them up front and invite questions from the audience. So first, Catherine Burns. Catherine, do you mind standing? Is a professor of systems design engineering and is the executive director of the Center for Bioengineering and Biotechnology. Wave to the crowd. She's also the director of the Advanced Interface Design Lab. Catherine's research is focused on understanding human error in hospitals. It's one of her research areas. So human error is a major cause of error in Canada. 1 in 20 challenges related to difficulties for patients results actually from human error. And in the US, it is the 3rd leading cause of death, so just behind cancer and cardiac causes. So the influence of human error in healthcare is not studied very well. We're very glad to have Professor Burns leading us in this direction and exploring new ways to reduce human error, and particularly in the use of technology to support healthcare. So today, in her mini talk, she will discuss current technological approaches in healthcare and the successes and the challenges, and also the difficulties associated with the ethical use of technology. Dr. Burns received a PhD in mechanical and industrial engineering and a master's degree in industrial engineering from the University of Toronto, and a bachelor's degree in systems design engineering right here at the University of Waterloo. She has co-authored over 250 publications and 7 books. She's currently leading an internationally recognized program of research in human factors engineering, and she was awarded a $1.65 million NSERC CREATE grant to establish a biomedical technology graduate program at Waterloo in collaboration with several other institutions. Catherine has received Waterloo's Outstanding Performance Award 5 times and is a fellow of the Human Factors and Ergonomics Society. Juan Liu is a chemistry professor affiliated with the Waterloo Institute of Nanotechnology, the Water Institute, and the Center for Bioengineering and Biotechnology. So you can see we have many realms of interdisciplinarity within each of our candidates here. His research focuses on chemical functions in biological systems, such as ligand building and catalysis, that are mainly realized by proteins. So in particular, DNA-based ligands and catalysts are very attractive, principally because of their high stability and also because of fiscal constraints. They are low cost. So today, Professor Liu will discuss his work on selecting these DNA molecules specific for various metal ions, small molecules, and microbes. He received his PhD in chemistry from the University of Illinois. Prior to his appointment at Waterloo, he worked for 2 years as a postdoctoral fellow at the Center for Microengineering Materials at the University of New Mexico and the Advanced Materials Laboratory in Sandia National Labs. He developed there a very novel drug delivery system. He has published over 200 scholarly publications. He is, at the University of Waterloo, a university research chair. Carolyn Wren is a professor in mechanical and mechatronics engineering. She's director of the Waterloo Microfluidics Laboratory. Her research focuses on microfluidics and lab-on-a-chip technology, and it has played a key role in developing innovations in advancing disease diagnosis and drug screening by serving as a very interesting and novel enabling platform technology. Her research presentation will outline her seminal work on lab-in-a-chip. We're all excited to hear about that, for protein fractionation and biosensing. Professor Wren is also works towards, wearable technologies. Is that right? So drug screening, single-phase microfluidics for wearable technology. Carolyn received her PhD in mechanical engineering from the University of Toronto. She holds a Tier 2 Canada Research Chair in Droplet Microfluidics and Lab-on-a-Chip Technology. She's a Fellow of the Canadian Society of Mechanical Engineers. She co-founded 2 companies related to protein separation for pharmaceutical applications and electrical sensing towards healthcare and environmental protection. She was recently named a member of the Royal Society of Canada's College of New Scholars, Artists, and Scientists. Roderick Slachev is a professor in the School of Pharmacy and cross-appointed to the Department of Biology. He's a member of the Waterloo Institute of Nanotechnology. His research focuses on the application of bacterial viruses and their genetic systems in the generation of novel therapeutic platforms. One particular platform has led to the production of a novel vector production system, DNA mini-strings, which is quickly becoming the international paradigm for future gene and cell therapeutic applications. Traditional gene and cell therapies on the markets are very, very off the charts dollar-wise, ranging from $400,000 to $1 million per treatment. They offer only a single application per patient and have massive variability between patients. They are difficult to maintain, scale, and store, which has contributed to up to a 2-year backlog in the industry. Today, Professor Slachev will discuss how the DNA mini-string platform is capable of expressing a multi-gene cassette of interest safely and affordably without compromising efficacy or efficiency of production. So Roderick received his PhD from the University of Saskatchewan and an MBA from the Biotechnology Management and Commercialization from the University of Saskatchewan. What an interesting combination. After completing a postdoctoral fellowship of the Department of Medical Genetics and Microbiology at the University of Toronto, and he also gained industry experience, Roderick joined Waterloo. He currently directs the School of Pharmacy's award-winning Business Curriculum Initiative and is the founder of Mediphage Bioceuticals Industry, which is a pioneering cell and gene therapy platform for personalized, redosable genetic medicines. So, a long introduction. I wanted to do it for all of them at the start so that we do not interrupt the flow from speaker to speaker. So please, let's first thank the speakers for joining us today and welcome them warmly. So the flow is I'll invite each of the speakers, and if we could just move along from speaker to speaker, that would be helpful. So no introductions between speakers. We'll start with Catherine Burns to tell us a little bit about her research. After that, we'll have the speakers at the front, and Bernie Dunker will moderate a question and answer. So keep your papers and pencils handy for questions that you'd like to bring to our panelists at the end of their presentations. Catherine?
Great. Thank you for the wonderful introduction. Okay, I've got 10 minutes here, and I've got 2 things to convince you of. Okay, and the first one is that the human factor is the biggest variable in our healthcare today. Okay, and the second thing is that we're perfectly capable of making the situation a lot better than it is. Okay, do you already believe me? Then I'd be done. Okay, so I think Charmaine mentioned and that's going to come up on the slides, is that human error is, by some counts, is certainly in the top 10, okay, of leading causes of death, okay, in hospitals and in healthcare. That's a terrible way to die, right? Wouldn't it be much better to at least die from diseases than from human error, right? Okay, so it's quite disturbing. The other thing that's actually really disturbing about it, okay, is that there's nobody out there who wants to make a mistake on you as a patient, okay? And I've got friends who are physicians, okay, and they have made mistakes, they have made errors. And what do you think that means to them? Honestly, it's life-shattering, okay? So I think that's something we need to understand as well, is that we've got people out there who really want to do their jobs absolutely as best as they can, and they need and they deserve our help, okay? So they deserve everything we can do. So this is a paradox that we're dealing with, okay? We're dealing with the fact that we do have remarkable technologies out there, okay? We have made remarkably significant and important contributions to the treatment of health, okay? We have a lot of sophisticated pharmaceutical treatment, okay? So there's an awful lot that we can do, and it's remarkably powerful, okay? And we've spent a ton of money doing it, but there's a place we haven't spent a lot of money, okay? And that's on improving that human factor, okay? And that's what I want to work on, and that's what drives me, and that's my passion. That's what I'm talking about today. And so the human factor, I mean, I talked a bit about the clinical side, human error by clinicians, but there's another part I'm going to talk a little bit today about as well, and that's about us as a patient, because healthcare is changing, okay? A lot of your healthcare ends up being your responsibility, okay? And one of the problems I'm working at right now is medication management, okay? I'm working on with my colleague Kelly Grindrod in the School of Pharmacy, and this is one of the issues that we're dealing with. Who actually administers your medications to you? Guess what? It's probably you most of the time, okay? Unless you're maybe in a hospital, somebody might give it to you, okay? So there's a lot of your healthcare which is in your hands. Do you do a good job of it? No, actually, probably you don't, okay? You guys might, okay? I'll give you the credit that you might, but the statistics say that 50% of patients will not take their medications as prescribed. So what does that mean? It means they're not going to get the benefit of that. So if we go back to that previous slide, I think it was 2.6 billion invested in pharmaceutical development. Kind of wasted if you don't take it, right? Kind of wasted. Poor adherence is a massively important problem, okay? And before I go on, there's actually some really good reasons why people don't adhere. Maybe they think they don't need it anymore. Maybe they're having side effects, okay? Maybe they're too busy. Maybe they forget. Lots of good and normal reasons, okay? Another example of a human factor that we're working on is fitness and exercise. Who gets the exercise that they know that they should? We know we're supposed to do it, right? Okay, but we don't get around to it. Only 2 out of 10 adults, these are the Canadian recent data, 2 out of 10 adults, 1 out of 10 children are meeting the physical activity guidelines. Does physical activity affect your health? Absolutely, you betcha, right? Okay, we know that. Okay, it is often thought to be the primary cause, preventable cause, okay, of modern disease, okay? So I'm gonna keep moving on, but there's a few reasons for this, okay? One of these is definitely that people are not very good about doing the actions that are needed to affect long-term problems. We see it in healthcare, we also see it in areas like environmental management and pollution, right? Those long-term decisions are very hard for us. If you want to learn more about that, you can read Kahneman's book on thinking fast and slow, okay? Exactly that problem. So what can we do about it? We do have that opportunity through technology to try to make this better, okay? In my lab, we study human decision-making. We've got very strong techniques for that, and we try to figure out how we can drive better decision-making by giving people better information, motivating them to take decisions maybe a little earlier than they need to, okay, and trying to push on that. Okay, so these are some of the projects that we're doing right now, okay? And you'll see that connection to the problems that I brought forward. Don't know what my time is like. But we are working to try to motivate people to increase their exercise behavior, okay? And we're looking at using mobile sources that can give them encouragement, that can provide them triggers, that can encourage them to kind of take that extra step. So if they're close to doing it, push them over to take that extra walk, make that extra step. That's one of our studies ongoing right now. We are working to help patients make better medication decisions, okay? Because we feel that if they actually knew what their medication was for, okay, if they knew what to expect with it. Okay? They might actually take it a little bit more. Okay? They might take it a little bit longer. They might get through that minor side effect, okay, and actually start getting the benefit of that. We've been looking at how to get healthcare workers to keep their data up to date. Okay? Data is one of the biggest things in healthcare these days, okay? But the quality of those data records can be extremely poor. Why? Because entering that data, okay, is not really a very fun thing to do. Who wants to sit around entering data, okay? So that's another project we've been working on, is how do we encourage healthcare people to enter their data in a timely and accurate kind of manner, a job they may not want to do, and how do we make that better for them? And we're starting projects looking at stress management. Okay, so how do we get people to understand when they might be under high levels of stress and where they might need to take a little bit of a break, okay, and move away from that. So does it work? Okay. Absolutely. So this is our study. We ran this last year, looking at clinicians at the Guelph Family Health Team, entering data. We gave them 2 pieces of information, okay? And we told them how they were, what was their percentage of timely data entries, okay? And then we told them their performance relative to their team on entering their data. Okay, and you can see from the chart, as soon as we did that, we had a massive performance increase, okay? It was very noticeable, very definite. How do we do this? Okay, we spent time understanding their decision-making processes. We spent time with their managers trying to understand what behaviors they wanted to influence, okay? And we spent time talking to our users, okay, trying to understand what were their motivators, okay? And this particular user group was very motivated by kind of an intergroup competition, okay? So they wanted to keep up with their peers, okay? And so that's one way we can start improving people entering their data. Okay, studies that are currently underway. We're currently looking at whether or not we can give patients, and this seems so silly, honestly, on their medications, the reason why they are being prescribed that medication, okay? This seems like the silliest study ever, but actually you're not really told that directly, okay? Or you might be told it verbally, okay? And you might be on that medication for years and you might not remember that. If you go to the doctor to get one medication, you probably know what it's for. Okay, it's for what you asked for. Okay, if you go in and you are 80 years old and you're on 5 to 8 medications, which is very possible, okay, you may not know what each of those are for. Okay, and that affects your ability to understand why you're taking that. Okay, it affects how well you're going to adhere to that treatment regimen. The other thing which is really important is it affects the kind of conversation you're going to have with your physician. And in a study that we've been doing on this, we've been talking to patients. Would you like to know what your medications are for? Talk about easy research. Yeah, they said, yes, we would. Okay? Would you like your pharmacist to know what your medication is for? Would you be willing to share that information? Yes, they would. I don't do a lot of studies where I get 100% results. Hey, this one we did. Okay? And you know, this is a group that's taking, on average, about 4 to 5 medications. Okay? So a kind of fairly complex group. Why would they take it? Okay, a lot of good reasons. So they can understand things better. Okay, so they can understand off-label uses. Sometimes you prescribe things for different reasons. Okay, that may not be obvious to you. You know, reducing errors. Okay, back to that statement at the beginning. Okay, about the prevalence of human error in medicine. Okay, a lot of those errors come from medication use, okay? Or from being prescribed the wrong thing at the wrong time, or for 2 things that are the same thing at once. Who catches that error? Who do you think catches human error in healthcare? You do. Okay, the patient catches it. Okay, so the more we empower this patient, the better information we give them, we're going to tackle that human error problem as well. Okay, so that's also kind of something I want to kind of bring forward. Okay. So where are the research questions right now for my work? Okay, definitely trying to understand what's that right information that's going to trigger your behavior to become more effective. Okay, we don't want to overload you with information, but we want to give you that right information that helps empower you better. Okay, what are going to be the right motivators? Okay, they're different in every context, they're different in every demographic. Okay, so it's a little bit tricky and the science is not clear yet on that. Okay, we don't want to gamify everything. Okay, we do that in some cases. But we're really talking about kind of triggering deeper motivations here. You know, can we personalize your motivators? When do we actually trigger a new behavior most effectively? okay? Because we don't want to do that in a way that interferes with the things that you're doing right now, okay? We don't want to take away from another behavior that you have to do. So knowing exactly when to make that triggering, okay? That's a scientific question for us in my group. And how does your workload change if I push on one behavior, okay? Have I taken away another behavior? Okay, have I done something I didn't want to do? So we really need to understand workflow, okay, and work processes to do this in a very well and scientific kind of way. So definitely the challenges are there. Okay, the sciences area is still fairly poorly understood. My group has a lot of strength in this area, okay? We are kind of world leaders in something called cognitive work analysis, okay, and that allows us to open up people's work processes with a lot of granularity and detail and try to understand where we can start pushing behaviors in really effective ways, okay? And then we use that and we build that into technology to move it forward. So that's really what we do. Yeah, and I hope that I've convinced you that human error is one of the most untapped areas, okay, of healthcare that we need to tackle. And as well that we can bring science and technology to bear on it, okay, and we can actually make this world a lot better through that. Okay, thank you.
Hey, good afternoon. It's a great pleasure to be here. So health and environment are among the most important problems we're facing today. Specifically, we want to improve biomedical diagnosis, therapeutics, environmental protection, monitoring, and remediation. As a chemist, a lot of them, to me, has to do with molecular recognition. So let me give you some examples. So this is glucose meter, pregnancy test. So for these very popular devices, the core molecules, right, for the detection are proteins. So for glucose meter, it's an enzyme called glucose oxidase, and for pregnancy test, it's an antibody. So in my lab, we want to use we want to test if we can replace these functions by nucleic acids, especially by DNA. The motivation comes from DNA being more stable, lower cost, and oligonucleotides can be chemically synthesized with very good batch-to-batch consistency, and during the synthesis, you can easily modify them chemically. So this field started really about 3 decades ago, and now it has gone a long way. So this is the structure of DNA, right? So we all know DNA is the hallmark for molecular biology. And chemically, if you look into more detail, right, it has got a phosphate backbone. If you open up the duplex, you expose the bases, right? So the base also has many chemical features that can interact with molecules. So here I give you an example of interacting with metal ions, right? Different metal ions can bind to different places in the DNA. And of course, the DNA can also bind to other types of molecules. So how do we evolve DNA molecules with interesting and useful properties? So here's one example. So in my lab, we construct a library like this, right? So on the 5' end of the DNA, we put our fluorophore. Then we design the library, so it's all DNA, right? Except here, we have a single RNA linkage. So RNA stability is lower than DNA, so that's an artificially introduced cleavage site, right? We want the cleave reaction to happen here. Essentially, we'll screen a DNA molecules that can do this reaction. So here we have 50 nucleotides. It's random. So each position can be A, T, C, or G, right? So 50 of them, we have the possibility of 4 to the 50s. So this is a huge number, right? So in the lab, we cannot make that many. We make about 10 to the 14th. This is still a very big number. We just hope within these many molecules, maybe we're lucky, we have 1, 2, or a few of them that is active. Right? So we can add a target molecule or metal ion in this case, and we hope, right, some is active, and if it's active, then the reaction can happen, right? this bond can be cleaved, and you produce a cleavage product. So if this reaction happens, then the library length will go down from 119 to 91. So with this length difference, we can easily separate them. So this part is what we need. The reason it can cleave could be because of this unique sequence, right? So that's what we are hoping for. So we harvest this, and then we can use a reaction called the polymerase chain reaction, or PCR, to amplify this. So that's the beauty of DNA. You can do these type of things with the enzymes available. So we can go back to the library by running 2 cycles of PCR, and we can do this over and over again. So that's called evolution in test tubes, right? So we start with very diverse, large population, and we give it some pressure. Only those winners can survive, and they make children, right? A lot of them, with the parent. So does this sound familiar to you recently, evolution in test tubes? Last week, right, we were very happy, excited about the physics Nobel Prize awarded to Donna. But what about chemistry, right? Here we go. Right, chemistry, so it's awarded to pioneering work, right, in test tube evolution for new enzymes and antibodies, right? We just said, no, these are proteins. So essentially what I just described is the same technology, but we are evolving nucleic acids instead of proteins. So this is the first work did in my lab in Waterloo here, 2014. We published that, and that's the library sequence, right? So we obtained this sequence. The blue region here is 15 nucleotides, and we optimize it, and we obtained this molecule we call C13D. So the bottom strand is what we call enzyme. It's just pure piece of DNA, right? It's no protein, it's just DNA, and it can catalyze the cleavage of the substrate strand at here. Once we add a metal ion, in this case it's a lanthanide ion. So if we put a fluorophore on the substrate strand and a quencher on the enzyme strand, once these 2 strands are hybridized, the fluorescence is quenched, right? So it's dark. When we have the metal ion, it can cleave here, and after cleavage, you have this very short piece that cannot stably bind to the enzyme strand. It will release. Then we produce a fluorescence signal, right? So by this, we can actually see this reaction very easily. Using the same method, we can obtain other types of DNA sequences. You can see here, the loop sequence is very different, right? So this piece of DNA can detect cadmium, right? So you can see here, only cadmium produce the signal, and we can detect down to very low concentrations. And similarly, we obtained another piece of DNA that can measure copper, right? So now you can see here, we have activity with copper, but cadmium is now silent because the DNA sequence here is different. That's one we obtained for silver, right? So it's a bigger loop, but now you can only see cleavage with silver, but cadmium and copper, they're both silent. So these are all what we call thiophilic metal or heavy metal, right? So silver, copper, cadmium, they are toxic, right? So we can also obtain DNA that can detect physiological metals such as sodium, right? So this piece of DNA, you can see here, is sensitive to sodium, right? Not lithium, not potassium, or other metal ions. And here's another example we obtained for calcium, right? So calcium, and in this case, actually used 2 calcium ions cooperatively. So we made this cover figure to draw scissors, right? So this has to act together for the cleavage reaction with very good sensitivity and selectivity for calcium over magnesium. So last year, we wrote a review paper published in Chemical Reviews to summarize the field, basically using DNA to recognize the elements, mostly metal ions. The green spots, more or less, right? So the DNA sequence is corresponding to them. So we hope one day we can cover the whole table, at least the metals, with DNA, right? Each element, we want to give it a DNA code. So this paper was published just last year and already received over 100 citations. So I think we're very pleased to see that. So DNA can not only just do metal ions. So I give you examples of metal ions because I love metal ions. And it can also do other things, right? So like these types of DNA sequences we call aptamers. Now that means they bind things like antibodies, but they don't do catalysis, they're just binding. So it's called aptamers. This type of DNA can bind, for example, ATP, right? That's our energy molecule. And there are also many DNA sequences evolved to bind to specific cancer biomarkers on cancer cells, right? So like for this piece of DNA, reaching guanine can do that. And we've been working with Ecosynthetics, which is a company in Burlington, and to attach our DNA to the starting nanoparticles for drug delivery applications. Recently, we've been working with Cranco, which is a small company in Mississauga, to try to obtain DNA aptamers for cryptosporidium oocytes. So these are waterborne passengers that can cause infections. So we want to be able to detect them in water as well. So we will continue to select new functional DNA, as I just described, not only for metal ions, but also for many other types of molecules and cells. We like to perform fundamental biochemical characterizations on them to understand them as enzymes and as ligand. So I've always been wondering, right? So are these type of DNA-based activities just the totally chemical functions, or were they actually once upon a time happening in nature, right? So the current world is dominated by proteins, right? The protein world, enzymes, antibodies, right? So this hypothesis is called the RNA world, right? So maybe a long time ago, the world is dominated by RNA before proteins even exist, right? So RNA is much simpler. And we're working on like this even older molecule, maybe DNA, right? So so that has something to do with origin of life, right? So with this, maybe what we're doing is to rediscovery of the lost functions of nucleic acids, right? They were abandoned by evolution, but maybe we can pick it up and do something useful with it. So my lab also works on lipids, right? So we make liposomes, and so that's a paper we published just recently on Langmuir to summarize our work on these inverse lipids. If we can encapsulate DNA, right, in liposomes, essentially we're making some protocells, and that may give us some very reduced physical systems for fundamental studies. Of course, we want to push it for applications, right, as I just said, for biomedical diagnosis and for environmental monitoring. So that's all I want to say. Thank my research group, some of them are here, and some methods assigned to different students, right, who have done the work. Many postdoc graduate students, and especially thanks to undergraduates here, very good quality undergraduates, right? So with our co-op program, I've recruited many of them, and also visiting scholars and students. Thank you.
Yeah, okay, thanks everybody for coming. And some of you have been familiar with what I've been presenting in other places. So I'm a mechanical and electronics engineer. So you'll see my slides. I'm not digging into that thing, more focus on developing innovations to help the other side, focus on help our other collaborator presenters here as well. So talk about the first one is for protein, and thanks to Jevin Liu was talking about the protein world. I'm glad to hear that. So this is early research about single phase microfluidics. What you saw from the top over here is we worked with a company called Converge Biosciences, where their life is doing the protein separation using capillary based. What we have done is we translated everything into microfluidic chips. I wouldn't be able to talk about all the details. Actually, lots of like human involved errors. This error is not really medical, but it's for the device. Here you don't see all of them. For example, take one particular example here. It's hard to see from here. You see the black dot here. What happens here is they have this capillary is about 100 micron, and they have a capillary beneath here is 100 micron. They have a membrane, which is hollow fiber. It's a kind of 100 micron diameter of the tube. Basically, what they tried to do, they did the Holman connections using glue. Basically, it's glue your hair to my hair and to another person's hair. You can imagine it's very difficult because if you put too much glue, it'll block it. If you put too less glue, it will have a leakage. So you're always going to have problem. So what you don't see here for here, we also glue, but we glue to use a machine and use devices, technology we developed. You see the performance of the here is it shows the protein separation side by side, and the top one is their system, bottom one is our system. It's for hemoglobin. Their performance is really good. Beyond of this, actually we developed company. We co-founded a company with one of my postdocs, Dr. Tianming Huang. The company is located in Cambridge right now. We founded it in 2010. We have been doing a lot of the work on the protein separation. Now we face the challenges here is, for example, here just to give an illustration, don't worry about all the details. Basically, our system is really good. It can separate lots of proteins, but for the pharmaceutical applications, especially develop cancer drugs, they're actually particularly interested in some of the small peaks because they could show some side effects or any other problems. Now, our hard time here is not only it's a hard time for us, it's a hard time for the protein separation community. Here's how are you going to collect this sample and put it into another system, for example, mass spectrometer, to understand what it is. It's very difficult because it's low abundance. Once you collect it and you dilute it, it's nothing. So it's there, but you cannot see it. So how can we do that? And now we focus on the improve the system, particularly say if I want to get this sample, by the end, actually our system, we think it collected, we just cannot see it. It's just so low abundant. We try to develop a new system where turn this one into another new company. That's what, not a lot of details, but here, what I want to mention here is leverage of the expertise of the mechanical engineering. What we play here, we play with the flow and heat transfer a lot. The whole focus on here is try to be able to collect this little guy as a high volume so that we can put it into mass spectrometer. We also develop a connection between our system to mass spectrometer directly. People can, GE Healthcare, the president in the Asian, she confirmed that this is a revolutionary technology, but we haven't completed the project yet, so know a lot of the talk. Second direction in my lab currently is we're doing this, I like the test tube. It's evolution in the test tube for the Nobel Prize winners. I call this mobilized test tube. What you can understand from here, this is a little drop. This is a water drop. In between of these water drops is oil. Oil as a carrier to move these tubes. My job here is really interdisciplinary. So all the people working here as a surface chemistry develop surfactant to stabilize the drops between the drops and the oil streams. And people use this for cell study, PCR, basically it's a DNA amplification, and do all kinds of things doing the organs. So my job as an engineer contributing to this field is how I can develop a system I can manipulate each of these drop as a test tube, robust test tube. Basically, I do whatever I can to manipulate this guy. For example, I say, okay, now stop, and it will stop. Now split this guy by 10% of that. Another one's 90%. 90% I'm going to throw away. That 10% has a key thing I wanted. Can I do that? Or I want to heat up this droplet. For example, I only heat up the first one. I don't heat up the second one. How can I do that? So that's the whole point I'm going to do here. So we're still moving along here. So there are a lot of good reasons we want to do this. For example, traditional system, you use a Petri dish, which is old school stuff. The very automated, the 96 well plate, 384 well plate, it's very robotically controlled, it's good. However, still have lots of limitations. So how many you can do by this? So this is, you go 96 well plate, you go 2 hours of, that's my collaboration with Professor Naka from pharmacy, and we do the drug screening for Alzheimer's disease. That's what his lab is doing. 96 well plate, run for 2 hours for one assay. How many assays you can run per day? You can calculate. Let it run for 24 hours. It's 12. So it's very difficult to do. However, if I compare to the slides here, if I can turn each of now what you saw this movie, it's kilohertz. I slow down the movie. Basically, you blink your eyes, I get thousands of the droplets generated. If I develop a technology, I can manipulate each of them as what I wanted to be a test tube. That means I can increase throughput kilohertz. For 24 hours, I get 1 billion. So that's the difference, what do we want to make. But still, it's a long way to go. That's why I'm here. The good thing about it here is all the droplets is equivalent to 196 well plate, but much, much, much smaller. The reason here is that this 96 well plate normally has a 200 microliter. Our little water drop like this is about 20 nanoliter. The benefit by doing this here is for the drug drug assay, what a person had told me, this assay, I need to buy all the chemicals, and mechanical engineer, we're not familiar with those chemicals. 1 milligram will come to my hand is $1,000 US. Oh, that's quite expensive. But anyways, so because we're doing these drops, we reduce this amount of the use of the reagent magnitude lower. It's 10 to 5 times lower. So that means that the cost of the saving is significant if I could turn this one into a good use. So now here just the illustrated, I won't go into all the details. Here's what I want to manipulate the droplet, but to do that, I need to understand how to generate droplets stably, how to track them, scan them, do whatever I want with them. So that's all the functionalities as an engineer I'm focusing on. Wouldn't go into the details. Basically here, I want to understand how I can generate droplet. So my tool can be used for biochemists. You don't have to have a PhD in microfluidics. You can still try this amazing platform, because my model will tell you how to design the chip in a simple way and a robust way. What I had here is the amazing thing I like it here is a lot of fundamental, don't worry about it. And we develop our model. Our model is so accurate. What you saw here is 108 set of conditions, including all the chemicals, geometric and operating conditions. Here's the experiments. If my model is correct, that should all collapse into one line, which is really good in showing on here. So in this case, our model is so accurate, we caught this stage, which had never been caught before, for generating this model. Before we started in 2000, I changed to this direction in 2010. 2009, there was a review article. Whatever the model available in the world, nobody agrees with each other, because lots of fitting parameters down to there. What we try to do is use a mechanical engineering understanding of all the force balance to cut off all these fitting parameters instead of bringing the theoretical argument into it. So that's why it's very accurate. Here just to show some glimpse of all the different functions. For example, in this case, I was used for the drug screening. What we had here is we have one chemical in here. I really don't want to give you the details about the chemicals, because actually I don't really know very much. So one side is the chemical, the other side is chemical B, and what my collaborator tell me is I need them to mix right now very quickly, seconds. We mix them very well. Like mixing index means that they're full mixing about 4 seconds. And then now we look at all these we put a drug inhibitor in there, see how they responded to this assay. So assay, please ask Professor Decker, I don't know. So now we move on to the sensing part. So I want to know where they are. And in the biggest platform where they are, I want to control the temperature, because lots of reactions are actually temperature controlled, temperature related. So in that case, what I would develop is microwave sensor. I'm pretty keen on this. This turned into my second startup company. What we want here is my goal here is I want to hit this droplet in a certain temperature. For example, I want to hit this droplet to 30 degrees. This is to 37. This one to 40 degrees. I cannot think about a better way to do. I'm pretty limited. If you have a brilliant idea, tell me. So I can think about a laser could do that, but laser to synchronize. Don't forget this droplet, now I slow down the movie, they come at kilohertz. It means every millisecond I get one. How I can respond to the system to do that very well, that's very difficult, because I need to know what it is and heat it up accordingly. So, but a microwave could do, microwave is what are the way we work with our cell phone, it's gigahertz. By nature, it runs very, very fast. No limitation for this kilohertz performance. So that's what we want to do. And also we develop a trigger here. Is basically this droplet, whenever droplet appears, it's a trigger to trigger my microwave. Now you're not only a sensor, you are a heater. So we can heat up to 70 degrees within 50 milliseconds. It's going on, it's almost simultaneous. We also turn it into a mixer. We kind of play a lot with that. Here, this is one thing is probably good to the pharmaceutical people is that we put the hydrogel materials down here, and when it goes through the sensor, this hydrogel material is heated up. They dislike the water. Now you see the water, the hydrogel beads formed over here. As an engineer, normally I want to measure temperature. I want to measure temperature, make sure the temperature is there. So we measured and said, okay, is it really true? Let's do an application. Application shows, yes, it's there. So that's really kind of cool. We're still on the way to answer all the questions, make sure our technology are robust enough. Now I move down to this wearable technology. I always think about microfluidics is the best way to do wearable technology. Okay, what are we going to do? We have lots of questions we could answer, but this one only one part of that. What we did is actually motivated by my student who got injured in the gym. What happens here is this Fitbit. I always wear this Fitbit. Fitbit does what? Fitbit tell you how fast, how many steps you walk, what is your heartbeat. Can they really help you? It really go down to Catherine's, can I go that motivation? It doesn't help me. Look at my weight. It doesn't help me. So what I was thinking about here is that my student got injured severely. 2 years he couldn't drive on the car. So what I was thinking here is, can I build multiple sensors over here? Probably now we actually only focus on knee, not everywhere. But I hope ideally down the road it will be everywhere. Can I put on a lot of sensor here so that I know before you go to injured, for example, you do this, and I was okay, I know what it is. My sensor will tell the brain will build up the brain. The brain will tell the actuators, so please, no, go back. So you need to give your force. But now it's a really interdisciplinary. I need to work with a colleague in the kinesiology, Professor Clark Dixon, is understand how many forces, how high the forces I need to apply. I can apply that, but what if I apply too much, actually injured on the other way? So that's the whole point we're focusing on. I'll show a preliminary concept. We're also kind of towards the funding a company on there. So this is my student who got injured. What I did is now this is a very preliminary study. It's a kind of a knee clip here, and it will show what the pressures are here, what is the forces we should apply simultaneously. This is very, very preliminary. I'm coming into this field, but I really see a significant impact in the world for the health problem. So that's what I focus on here. I think my time probably run out. Here is I just want to show the applications about this droplet, because I talk about droplet, but still probably doesn't sound real to you. What we work here, we work with Professor Krull from U of T, and his life is doing develop biosensing. What biosensing really means, he's ambitious. Put it simple. He wants to decorate this nanoparticle, one side with one molecule, the other side with another molecule. This nanoparticle is 10 nanometer. I was talking about glue my hair to your hair, which is 100 micron, but now it's a 10 nanometer. Yeah, he has a hard time to do in the traditional system. I said, you know what, I can help you. What we try to do here is we will encapsulate it in the little drop. This is a quantum bead. We put a magnetic bead first. Magnetic bead has 1 micron diameter. Now we put all the quantum dots onto the magnetic bead. For example, what you see here, this bright spot, magnetic bead doesn't shine, but now it's quantum dots all sit on this. How they can sit on this nicely is because this droplet actually inside has rotating vortices, help all the quantum dots go down to the magnetic bead. But there's chemistry in there. They modify the surface, which I don't know. So in that case, we develop on this. Now we have a long way to go down, but here just another glimpse about that drug screening. That's, remember I talked about 2 drops put together. That's drug screening. Here's when we make the hydrogel beads. The hydrogel beads are really, really uniform. That could be down to the pharmaceutical application. I'm still looking for the real applications, really push down to application. I'm not 100% sure yet. So far, we're trying to build collaborations and develop technology. Thank you very much.
Does it work? Okay, here we go. Anyway, first of all, that is some amazing stuff. Really prestigious group, and it's really an honor to be here. I have to say, you know, I had that song from Sesame Street going through my head, and like, one of these things doesn't belong here. Anyway, I'll do my best to keep up with something that might be intriguing. As mentioned earlier, everything that we do in our lab is really based on the use of bacteriophages and applications that come from bacteriophages and in the development of new therapeutic platforms. And the reason why this is so intriguing to me is that bacteriophage really form the largest genetic reservoir on the planet. So they're massively untapped in terms of the types of genes that could be applied toward therapeutics, as well as genetic systems that can be retrieved from them. And a lot of what I'm going to be talking to you today about is one such application that comes out of my lab and that we're working on in a few labs at the moment towards a new type of gene therapeutic system. And I say gene therapeutic system because I don't like the word gene therapy or cell therapy. And the point here is to really kind of change the entire process and move to something that we like to refer to as genetic medicine. Oh, sorry. So to start off, I wanted to provide this as a line that came out of a 2005 paper in Nature, that the concept of genetic medicine is extremely powerful. But as powerful as it is, it's also very dangerous. And there have been a lot of issues and a lot of dead people that have come through a lot of the clinical trials and new applications in trying to build what would be gene or cell therapy. And as I mentioned, this is something that has to be taken into account, and we need to move away from the concept of what is just traditional gene therapy, and we need to move towards something that is genetic medicine and specific and personalizable for individuals. So here are the kind of landmarks that you would see for any gene type therapeutic or cell therapeutic. Okay, so it has to be safe, of course, has to be effective, or else there's no point, and it has to be accessible. So there are two main streams that come here. The first is the viral stream, and the second is the non-viral stream. And each has a kind of trade-off that comes with it. Viral systems are, because we've co-evolved with them for millions of years, they're very effective at getting and delivering genetic cargo to cells. They can also be very specific for where they're delivered. On the other hand, safety is generally an issue because we've co-evolved with viruses for millions of years, so the body also knows how to get rid of these. And in terms of accessibility, viral vectors, particularly lentiviral and AAV, have a backlog at present of about 1 to 2 years. So that's not really acceptable if we want to push forward where we can go in genetic medicine, and we need a new type of system. And they're also extremely expensive. So we have also another important part when we look at the effectiveness of viruses. They may be highly effective, but how do you make a one-size-fits-all? Right? And this is an issue because you may have come across AAV in your life or potentially a lentivirus. And if you're trying to use this as an actual vector for any type of genetic cargo delivery, well then it may not work for you. On the other hand, that same dose might become highly toxic to somebody else, especially if it's administered systemically. So it's very difficult to find what is that initial dose because there's 5 to 85% difference in serotolerance between individuals. But more importantly, you can only get the one shot. So if I tell you about certain retinopathy treatment like Luxturna, you may be able to get a dose, and it might give you some vision that you didn't have before, which is rather miraculous, I agree. It might last for anywhere between 1 and 7 years. It might be better in one individual versus another individual, or it might not do anything at all because you've already been exposed. And after that, you're done. That's it. You don't go back and try it again because you built up an immune response to that same virus. So that's a real issue. Now on the non-viral side, yes, it's safer. The problem is that it sucks. It doesn't work. So in terms of the actual transfection efficiencies that come with non-viral vectors, they tend not to work very well. Very low cellular uptake. And there's also a potential for insertional mutagenesis. This goes for any type of DNA that makes its way to the nucleus. And the longer the durability, the longer the chance of an actual insertional event. And if something inserts into an oncogenic suppressor or upstream of a proto-oncogene, well then you can actually end up with a malignancy. So you can be trying to treat cancer and actually cause cancer. And that's not okay. And that has happened, and several people have died. Just recently in a Juno trial for CAR T, there have been 18 deaths that were associated with that trial. Sorry, I'm just a little winded from a little food poisoning that I'm getting over from yesterday. So there is also an increasing regulatory stringency that comes with these scenarios, and we need a newer system that really allows us to move away from these and captures all of these. They have to be captured or there's no point in proceeding forward. And that has not been done until, we think, now. So, it's funny, when I look at this, this is the actual vector that we produce. And the most important part of this vector is that little SS site that are on the two ends. And that's what allows for its production, also allows for nuclear translocation, and all of the enhancements that we see with it, also very important in the actual topology that's generated by that system. So to put it simply, I've spent the last 10 years developing 300 base pairs, which is rather sad, I think. That being said, it's the modular component of this which is the most important. We can do whatever we want with it. You can control the promoter. You can control what transgene, of course, that you want to deliver. You can control the durability. You can control the level of expression, the specificity, the ability to nuclear translocate into any type of cell. And that provides a lot of power then to what you can do with genetic medicine. More importantly, it's a minimal vector. So there's nothing on here. There's nothing on here which is going to cause any immunostimulation. And you do get that from traditional plasmid vectors even. And very importantly, if you actually have a translocation, or sorry, an insertional event into the chromosome anywhere, you're going to kill that cell. I'm just going to show you a kind of schematic of that. So it's safe, it's actually effective, and in terms of production, it's massively accessible because we can generate bucket loads of this stuff. So, as a gene delivery vector platform, it unlocks the potential of what we like to call personalized, redosable genetic medicine. If we get rid of all the immunostimulatory components that occur on any type of a DNA vector, we now have a naturalized biologic. So for instance, that same example that I was giving you before, like Sterna, imagine if I could give you as much as you need until we titrate you up so that you see, and we actually cure your disease. And you don't just get one shot at it. You get shots for the rest of your life as much as you need on an ongoing basis so that you can now see. And that may be different for you as it is for somebody else, but you'll get what you need to make sure that you actually now are looking at genes as an actual medicine that's redosable, titratable, and personalized to what your personal needs are. So where are the applications here? Applications, of course, in what is traditionally known as gene therapy. In oncology at present, where we're operating monogenic diseases, of course, cardiovascular disease, DNA vaccines, orphan diseases. And I'll be honest, I didn't even know about these. It's more companies that came to us because they wanted applications in those areas. Then, of course, cell therapies, right? So ex vivo approaches for CAR T, chimeric antigen receptor, or pluripotent stem cell, or stem cell itself. Even others that we're just coming across right now, like recombinant AAV production. And then, of course, gene editing. You're all familiar, I'm sure, with CRISPR-Cas9 and TALENs and other types of approaches. And this is a much safer way to be able to provide that type of gene editing. So I can't actually show you the data because I don't have time for it. So I want to show you just schematics of how this looks and why it's better. So on the safety side, as I mentioned, there's a prevention of an actual insertional mutagenesis. The topology of this vector, being linear covalently closed, is that if it actually inserts anywhere into the chromosome as an illegitimate recombination event, it pulls in the brake, and it actually kills those cells. That's not a common event, but you have to protect against it if it does happen. And when you're talking about any type of lingering DNA in a nucleus, the potential goes up with the increased durability as well. So you need protection against this system, and this has killed in the past. So we do need a kill switch built into any type of vector that we have that prevents against it. Secondly, effectiveness. What we found is that we're generally about 7 to 8.5 times higher in transfection efficiency for any type of the cells that we've tested. There is more data or more research that needs to be done here, but it's a good start. So we're not compromising effectiveness. And lastly, the accessibility. I haven't shown you the actual schematic here. But in terms of accessibility, we make this in E. coli. And as a result of it, we just found out recently that for a 3-liter batch in a bioreactor of E. coli, we can generate about 750 milligrams of msDNA, which is a lot, and that's quite a bit. So if we were to scale that up further, we can get into the gram level, and in terms of accessibility, that is no longer an issue then for the future of genetic medicine. So at present, there are currently about 2,600 gene therapy clinical trials. 13 of these have now been approved. There have been deaths that have occurred, as I mentioned, for Juno and a variety of others ever since early 2000. And most of these trials still work in the viral space. And this is amazing to me because lentivirus itself has been responsible for many deaths, and since 2000 hasn't really changed as a vector. And yet we still continue to use it. And that, to me, is absolutely amazing. And I think that that really is something that needs to change moving forward. That being said, this is a very, very strong and fast-growing area. This is the time for gene and cell therapies, or what I like to refer to as genetic medicine. There have been a number of acquisitions that have already occurred, and there's a huge, huge market that is developing and burgeoning around this. So if anyone's interested in that area, I would highly suggest that you apply your applications to it, because this is certainly the right time. And I think I'll end there.
Thank you so much. Wow, what for amazing presentations. Thank you so much for highlighting the robust research going on in the health area at Waterloo and the fantastic interdisciplinary nature of it. Fantastic. So we've got now a few minutes that we can devote to Q&A. So what we're going to do is we're going to pass a microphone around. We're recording this session, so it's important to have the microphone if you're asking a question. I'm going to ask the 4 speakers to come up. We're going to put some chairs up here for you, and we'll pass around a mic for your answers. So I'll give someone the mic to take up with you. There, Carolyn, I'll give it to you. Be the mic bearer for now. That's right. Okay, great. And while we're getting set up, just a shout out to the Center for Biotechnology and Bioengineering that actually runs a regular biomedical discussion group. And I had some involvement with that in the early days. It's been very well run. So if you enjoyed the talks today, check out that series through CBB. All right, so questions for our speakers. Yes, Scott. We'll just wait till you get the microphone. Yeah.
My question's for Roderick. Your vector system, I think if I heard your presentation or understood your presentation correctly, your vector can be used as a replacement for gene editing, such as CRISPR-Cas9 system?
Yes,
that's
right.
Is it synergistic with that, or is it actually a replacement for that?
It's not a replacement in terms of doing the actual gene editing. It's just a platform in which to be able to deliver the necessary genes for either of those systems.
I see. Okay.
Yeah.
Interesting. Have you done much work in that? Have you explored that much?
A lot of this is not actually being done through us. Our interest is really on the therapeutics and a few oncology indications at present. But as I mentioned, there have been a number of other companies that have come forward with their interest in it. So Caribou, for instance, would be one of them, CRISPR Therapeutics. And they have interest in sort of applying a technique like that. Because again, it's a much safer for them. I mean, they don't want... It depends what you're using the application for. In that case, durability is not something you're after. You want a high dose, you want to get the actual editing done, then you want those vectors gone. So having a protective mechanism like that in place is something that's of interest to them.
Yeah, interesting.
So I actually have 2 questions, and I hope they're both short. First to Catherine, I was astonished and quite frankly taken aback by your statistics about how many human-based errors in hospitals account for deaths. And you mentioned that doctors do make mistakes and that they feel badly about it, et cetera, et cetera. I understand all that. But are these errors, these human errors that are made by physicians, for the most part judgmental errors, or are they black and white errors? Because if they're judgmental errors, where you're assessing a set of data, and you assess it to some extent subjectively, to some extent that's understandable. So I'm curious to know what proportion of these errors lie in the realm that could be forgiven, if you will, versus the realm that perhaps should not be forgiven.
Okay, so I guess I'll just start from my perspective. Whenever a story ends with human error, that's the beginning of the story. So I believe that everybody's trying to do their job as well as possible, and all errors should be forgiven, and it actually indicates weaknesses in our system. I haven't really seen that particular breakdown, John, on what type it is, but a high percent of it is medication errors. There's issues with look-alike, sound-alike names, with misappropriate prescribing. And I actually think they're certainly not intentional by any means, okay, but they're working in an environment with a lot of complexity, okay, with inadequate support. There's a lot of comparisons made on the human error side between healthcare and aviation, okay, and that's an area that my field looks at a lot. And aviation is highly regulated, and they have a very unique kind of reporting system where actually they're encouraged to report their mistakes. And what we see in healthcare is entirely different. We see a litigious environment where they're encouraged to hide their mistakes. And what we see in aviation, which is even more important in some ways than reporting a mistake, is they are encouraged to report near misses. So they're encouraged to report that moment where like, oh my goodness, I almost did that. And that encourages a discussion, okay? And it brings that community together. And we're not seeing that in healthcare right now. And I think that's a really important direction for us to be moving on.
Carolyn, I just wanted to add to John's question, which I think is a really good one, and your answer is a great one. But just to tie in a little bit more to John's statement, is that, you know, when you evaluate, for example, surgeons or physicians on errors that are made and patient care, one big factor that needs consideration is that sometimes the best doctors are the ones that get the hardest cases.
Exactly.
And, you know, so ranking them by errors, etc., you need to take into account that this guy is the greatest, and he's going to get the toughest cases. And so sometimes, you know, it's tricky to navigate whether he's made a cut in the wrong place or
That's right. Yeah.
prescription. So just throwing that in as well.
Yeah, you do have to understand they're never working with perfect people, right? They're always working with people who are injured, sick. And definitely the harder and more challenging that is, the more challenging their situation is. So the context is very important. Yeah.
So if I may be allowed one more question. Roderick, I very much enjoyed your presentation. I think your mini-string DNA has huge application, but I'm a little concerned about the kill switch and the following possibility, and maybe I'm wrong, but how do you deal with the very real possibility of selective insertion, and from that, the possibility of selective killing of cells, accidentally because it's insertional, such as, for example, reproductive cells or another subset of cells that are absolutely crucial for life and for reproduction?
Yeah, that's a great question. The best answer I can give you is that, I mean, when you're looking at these types of insertion events, particularly like illegitimate recombination events, they're not common. So whether they happen at somewhere in the sort of 10 to the minus 4, 10 to the minus 5 frequency, when you're talking about billions of molecules that you might be adding to one individual for any type of a therapeutic, then you just need to be able to account for those types of situations. And if it were to happen, you have to make sure that a cell is removed from the population. But in and of itself, it would never have any useful application as an actual cell cytotoxic type of an approach. And it is something that we originally were thinking about, and an area we're moving in now is that for looking at recombinational events, for actually gene replacement, it might have a good role to play, because if you don't get the double crossover gene replacement homologous recombination event that you need, then at least you know that you remove those cells from the population. But by itself, it's not cytotoxic enough to cause any damage. Does that make?
Hi. So from what I understood from your talk, a lot of your focus is on physicians. How do you ever take into consideration where in kind of the chain of care those errors exist? You said look-alike, sound-alike medications, which sounds to me like an error, not from the physician side, but maybe from further down the line in, say, nurses or even the clinic sending up.
Yeah. Your question is well taken. And we don't do all of our work with physicians, so some of our work with pharmacy now has been with pharmacists, okay? We have a very complex care environment where not only, the physician is not the only person who provides your care. We have a whole bunch of healthcare professionals. The lines of communications between these professionals are almost non-existent, okay? It is one of the few areas where people are trained in totally different educational institutions in isolation from each other. They work on totally independent information systems, and that's one of the big issues that we're dealing with. But definitely the patient is becoming very important to us, and so some of our study with the medication, the reason for use, is really driving that patient. There's a movement in healthcare right now for shared decision making. That means bringing that patient into that conversation with that physician or that other healthcare worker, where they can really participate in choosing the treatment that's appropriate for them, works with their lifestyle and with their goals. But that requires, a whole bunch of different changes. It requires a change to how the physician looks at that patient, but it also requires us to develop an informed patient, okay, and have that patient empowered to have that conversation. And so that's one of the things that we're looking at as well. So it's a really, really important part. And I think patient empowerment is really going to be the future in healthcare in some ways, because I think everything gets downloaded, and we're going to be picking it up at the end, right? Yeah.
So I think we have time for one more question if someone else would like to ask one of our panelists. I see a hand up there. Sorry. Yeah. Or was there someone over here?
Thank you. My question's for Roderick. I'm curious as to your internal programs. What is the, maybe you can talk about your oncology internal program and how far along you are with it.
Sorry, we have a new oncologist that sort of joined our group recently. We're highly collaborative, so a lot we do is with other companies as well as internally with other researchers that are here. My oncology background is not quite that strong, so I'm not sure I could pull that off by myself. I'm more of a platform designer and more of on the genetic side and anything to do with phage. The interest that we have right now and what we're doing at Illumina is next generation sequencing to try to identify new markers for Alzheimer's disease as well as colorectal cancer and ovarian cancer. So that's kind of where that is pushing in our own direction at the moment. But even once we do find those, or hopefully once we find those, then I always look to others to try to collaborate, whether internally or externally, to try to move those forward. And that is, I guess, our vision, but also the nature of how we do things. We are highly collaborative in that respect. And honestly, I like that aspect of how to do science, so it works for me.
Great, thank you very much. Great discussion, everyone. Thanks so much. So this brings to the end of our session today for the Research Talks Forum. I'd like to thank our panelists again, Carolyn Wren, Catherine Burns, Joanne Liu, Roderick Slavchev. Thank you so much. I'd like to thank all of you for joining us today, and I'd like to let you know that the next Research Talk session is on December 5th. The topic is pushing back the frontiers of supercomputing, so I'm looking forward to seeing you then. Thanks again.